Okay. First question number 1A, part 1. What is sustainable food production? This question, I made this question from your lesson 1. These points should be included. First one is the production of food. Okay, the production of sufficient amount of food. Okay, the production of sufficient amount of food for the human population. For the human population. For the human population, using environmentally safe methods. Using environmentally safe methods. Okay, it is called as sustainable food production. So these points should be included. The production of sufficient amount of food for the human population using environmentally safe methods. Okay, environmentally safe methods, very important point. Environmentally safe methods. Okay, production of sufficient amount of food for the human population using environmentally safe methods. It is the sustainable food production. Mention 3 steps based on biological knowledge that can be used for sustainable food production. First point, production of high yielding varieties of plants and animals. Okay, first one is production of high yielding varieties of plants and animals. I think you have already written this the answer. If no, please search your resource book. These answers are available in your resource book under the lesson 1. Okay, first one, production of high yielding varieties of plants and animals. The second point, production of disease-resistant plants and animal varieties. Production of disease-resistant plants and animal varieties. Third one, improve the post-harvest technology methods. Improve the post-harvest technology methods. What do you mean by the post-harvest technology? After harvesting. Okay. After harvesting, what about the transport? What about the how they are arranged in racks in their containers? Okay, how it is done? It is the post harvesting technology. ඒ කියන්නේ crop එක harvest කරාට පස්සේ දැන් paddy නම් මොකද්ද ඒක crop කරාට පස්සේ ඒ ටික කොහොමද transport කරන්න අදාළ තැනට නේද ඕනම එකක් අපි හිතමුකෝ potato කියලා potato harvest එක ඒ කියන්නේ potato එකේ potato ටික crop කරලා ගත්තට පස්සේ අදාළ plant එකකින් කොහොමද potatoes ටික shop එකට යනකම් අරගෙන යන්නේ නේද එන්නේ ඒ ටික තමයි post harvest කියලා කියන්නේ post harvest period එක so post harvest technology improve the post harvest technology methods there are 3 steps. Name the type of the tetrose known you. It is the erythrose. Erythrose. That means C4. C4. That means tetrose. Okay. Erythrose are rare in nature. Erythrose sugar is rare in nature. Mention 3 works of cilia and flagella. First one, act as locomotor appendages. Act as locomotor appendages. Second one, can move fluid over the surface of the tissue. Can move fluid over the surface of the tissue. Third one, cilia lining in oviducts. Cilia lining in oviduct. Okay. Can you imagine if this is the ureter? Here the oviduct. Okay. So cilia lining in the oviducts. Cilia lining in the oviducts help move an egg towards the ureter. This is the ovary. Ovary produce the egg. Okay. This is the oviduct. This is the uterus. So these cilia helps to move these eggs towards the uterus. Cilia lining in oviducts help move an eggs towards the uterus. What are the components of the plasma membrane? It should mainly lipids and proteins. That enough. carbohydrates are also involved. So what are the components list? So you can write lipids, proteins, and carbohydrates. The main lipid type is phospholipids. Other than that, cholesterols are also involved. Okay, proteins and carbohydrates. What are the peripheral proteins of the plasma membrane? So how these three organic components are contributing the making of plasma membrane? Those are phospholipids. Okay, so lipids are involved. This is the phospholipid bilayer. Okay, protein molecules are embedded through this phospholipid bilayer, and again we can see protein molecules and carbohydrate chains. Okay, these structures called as glycolipids because this carbohydrate chain is attached to the lipid molecule, phospholipid molecule, and here the carbohydrate molecule is attached to the protein, therefore glycoprotein. Okay, and between these two lipid layers, cholesterol molecules also can be seen. So lipids, both are lipids, cholesterol, lipid and phospholipids. Okay, lipid, proteins, mainly these transmembrane proteins, and other than that, if this is the inside, okay, inner environment of the cell, okay, if this is the inner environment, nucleus and everything is there, so we can see peripheral proteins like this. Okay, peripheral proteins. Those are the peripheral proteins. You have to define what are the peripheral proteins. Proteins which are not embedded in the lipid bilayer. You can see these proteins are not embedded within the phospholipid bilayer. Proteins which are not embedded in the lipid bilayer and loosely bound to the inner surface of the membrane. Loosely bound to the inner surface. Okay? This is the outer surface. Okay, this is the outer surface and this is the inner surface. These peripheral proteins are loosely bound to the inner surface of the membrane called peripheral proteins. How we can define proteins which are not embedded in the lipid bilayer and are loosely bound to the inner surface of the membrane called peripheral proteins. Okay, then what are the three types of junctions found in animal cells? First one, tight junctions. Tight junctions. Tight junctions. Second one, desmosomes or anchor. Then desmosomes or anchor junctions and gap junctions or communicating junctions. Okay, first one, tight junctions, desmosomes or anchor junctions, gap junctions or communicating junctions. What is the active site of an enzyme? Okay, if this is the enzyme, always enzyme is larger than the substrate molecule. If this is the substrate molecule, the specific site, this site within the enzyme we call as the active site. This is the active site. Okay. The substrate binds to the specific region of the enzyme. The substrate binds to a specific region of the enzyme. This region is called as the active site. So that's enough. What is the cooperation? Cooperation under the enzyme part, what is the cooperation? Okay, what is the cooperation? Described in the allosteric regulation of enzymes. How we can define it? So you have to write this manner. Binding of one substrate molecule can stimulate binding or activity at other active site, thereby increase the catalytic activity. It is the cooperation. What is the cooperation? Described under the allosteric regulation of enzymes. Binding of one substrate molecule can stimulate binding or other active site, thereby increase the catalytic activity. It is the cooperation. What is the respiratory quotient? Respiratory quotient means, RQ. RQ is equal to VCO2 over VO2 now. So how we can define it? The ratio here, the ratio of carbon dioxide. Ratio of carbon dioxide evolved carbon dioxide and absorbed oxygen volume. No? The ratio of carbon dioxide evolved and the volume of oxygen consumed or absorbed. Okay, volume of oxygen consumed in a given time for the respiratory substrate. It is the respiratory quotient. The ratio of carbon dioxide evolved and the volume of oxygen consumed in a given time for the respiratory substrate. It is the respiratory quotient. What is the global importance of photosynthesis? Answer is available in your resource book. First one, all life on earth. All are depends on the sun. No? Sun, that means the photosynthetic organisms, that means photosynthesis directly or indirectly. So all life on earth depends on photosynthesis. Directly or indirectly. Second one, fulfill both carbon and energy requirements. For organisms. Third one, provide oxygen for respiration. Of aerobic organisms. Fourth one, maintain oxygen and carbon dioxide balance in the atmosphere. Fifth one, production of fossil fuel. Sixth, maintain the global temperature, because photosynthetic organisms absorb carbon dioxide. Carbon dioxide is the main reason for the atmospheric increasing of the global temperature. So photosynthesis maintains the global temperature by absorbing carbon dioxide. Okay, all the life on earth depends on the photosynthesis, directly or indirectly. Fulfill both carbon and energy requirements for organisms. Provide oxygen for the respiration of aerobic organisms. Maintain oxygen and carbon dioxide balance in the atmosphere. Production of fossil fuel. Maintain of global temperature. Which animal kingdoms have an organized nucleus? I ask about eukaryotic kingdoms. Okay, eukaryotic kingdoms. You know, there are three domains. Domain, archaea, domain, bacteria. These two are prokaryotic organisms. That means their DNA is naked, okay, exposed to the external environment. That means their genetic materials, DNA or RNA, is not enclosed by a membrane. Okay, they are naked. They are open DNA. But in domain eukarya, they have an organized nucleus. Nuclear materials are enclosed by a membrane. Okay, nuclear materials are enclosed by a membrane, so they are eukaryotes. What are the four kingdoms? Kingdom Protista, kingdom fungi, Kingdom Plantae, Kingdom Animalia. Kingdom Animalia. Okay. Name 3 protists which have heterotrophic nutrition. It should 2. According to your syllabus, 2 examples are enough. So, what are the 2 examples? Paramecium and amoeba. Okay. Heterotrophic means they can't produce their own food. They have to depend on other organisms' bodies or other organisms' productions. So they are heterotrophs. What are the 2 heterotrophic protists? Paramecium and amoeba. Under the protista kingdom, we are talking about algae. All algae are photosynthetic. That means all algae organisms are autotrophs. Okay, autotrophs. What are the similar characters of Neem to the angiosperms? You know about the plant classification. Up to now, we so many times we have discussed about this plant classification, how plants are developed, okay, how they become advanced. So under the presence or absence of the vascular system, mainly plants are divided into 2: non-vascular plants and vascular plants. Non-vascular plants and vascular plants. Non-vascular plants commonly known as bryophytes. And under the bryophytes, we are talking about 3 phylums. Phylum Bryophyta, example, Mosses and Pogonatum. And phylum Anthocerophyta. Example is Anthocerus. Third one is phylum Marcanthophyta. Okay, example is Marcanthia. Okay, these 3 phylums haven't a vascular system like xylem and phloem. No vascular tissues. That means they are non-vascular plants. And also in their life cycle, gametophyte is the dominant one. Okay, it is very primitive character. Gametophytic generation, dominant means very primitive character. In all 3 phylums, the dominant plant in their life cycle is the gametophyte. But they become vascular plants. Always its life cycle, the dominant plant is the sporophyte. Okay, sporophytic generation is the dominant generation. So it is an advanced feature. In vascular plants, we divide these vascular plants into 2 categories. First one is what? Seedless plants and seed plants. Okay, seed plants. Seedless plants, we are talking about 2 phylums. Phylum Pterophyta, example is Nephrolepis. And the second one is phylum Lycophyta. There are 2 examples you have to study: Lycopodium and Selaginella. Okay, what about the spores? In here, all bryophytes, homosphoric spores are presented. Homosphoric means their external feature is same to each other, but they are differ in the genetic composition. So homosporous, all are homosporous, all pterophytes are homosporous. Under the phylum Lycophyta, Lycopodium is homosporous, but Selaginella heteros. Heterosporous, that means megaspore and microspores. There are two different spore types, megaspore and microspore. In seed plants, okay, in seed plants all are heterospores, and seed plants can be categorized into two. First one is gymnosperms. Second one is angiosperms. Okay, angiosperms. Gymnosperms are three phylums. Phylum Cycadophyta. Cycus is the example. Phylum Coniferophyta. Pinus is the example. Phylum Neotophyta. Neatum. Here the neatum. Neatum is there. Okay. So they are evolutionary. There is a close evolutionary relationship between the phylum Neotophyta and phylum Anthophyta. Okay, here Anthophyta. Angiosperms means phylum Anthophyta. That means flowering plants. Okay, there is a close relationship between phylum Neotophyta and phylum Anthophyta. So there are some features, okay, which can be seen in neophytes related to the anthophytes. Okay, so what are the two similarities? What about their leaves? In neotum, its leaves look like this. Okay, that means their leaves looks like a leaf of dicot. Dicot means phylum Anthophyta, class Monocotyledone and Dicotyledone. So monocot and dicots. Here the neophyte leaf is external feature looks like a dicot leaf. It is the first one. External appearance of leaves are similar to dicot leaves. Okay, external appearance of leaves are similar to dicot leaves. Second one, they are seeds. They have no fruits. No, gymnosperms have no fruits, but angiosperms have a fruit. Their seed is enclosed by a fruit neck, but even they haven't a fruit. They are seeds. Their seeds look like a fruit. Okay, so the second feature is their seeds are look like as fruits of angiosperms. Their seeds are looks like the fruit of angiosperms. Tabulate the differences between two classes of angiosperms. Here, phylum Anthophyta can be divided into two classes, class Monocotyledone and Dicotyledone. So what are the five, I think, 7 differences, okay? 7 differences. Answers are already already available in your resource book. So anyway, we have to discuss rapidly. Here, monocots and dicot. Okay, what is the first difference? First one is the embryos have only one cotyledon. One cotyledon. Here, two cotyledons. Okay, the embryos have only one cotyledon. Embryos have two cotyledons. Embryos have only one cotyledon. Embryo have two cotyledons. Second one is, that is why monocot. Okay, cotyledon, like cotyledon. Therefore, they called as cot. Mono means one, here di means two. One cotyledon means monocot. Two cotyledon means dicot. Okay. So the second one is, what about their root system? Here, fibrous root system. Okay, fibrous root system. Here, taproot system. This is the taproot. This is the ground level. This is the taproot, and it is branched into two, and again branched. Okay, so many times branched. This is the taproot system. So, what about their veins? Veins in leaves here in monocots. veins are parallel to each other. Okay, parallel veins. Parallel veins, and here in leaves, the veins are reticulate. Reticulates like taproot system like that. Their veins are branched and again branched into two, again branched into two. This one also branched into two like that. Okay. So reticulate vein, reticulate veins. Okay. First difference is the embryos have only one cotyledon. Here embryos have two cotyledons. Fibrous root system, taproot system. Parallel veins in leaves, reticulate veins in leaves. Here, flower parts are trimerous. Trimerous means especially petals. Okay, trimerous means three petals. Okay, if you can observe a coconut flower, very small, but we can observe it. Only three petals. Okay, so trimerous. Here, pentamerous. Pentamerous, pentamerous or tetramerous. Penta means 5 petals, tetra means 4 petals. Pentamerous or tetramerous. Okay, flower parts are trimerous. Here, flowers are tetramerous or pentamerous. Here, perianth. Perianth present in flowers, that means no distinct calyx and corolla. Here, distinct calyx and corolla in flowers. That means, actually, this is the calyx. And what about the petals? In shoe flower, we can differentiate the corolla and calyx. But in coconut flower, we cannot separate these things. No, they are attached, like attached. So, how we can say it? No distinct calyx and corolla. Here, no distinct calyx and corolla. That means perianth present in flowers. Here, distinct calyx and corolla in flowers. Distinct calyx and corolla in flowers. Here, pollen grains are with one opening or aperture. One opening. Pollen grains are with one openings or aperture. Here, pollen grains are with three openings or three apertures. Okay. 7. Vascular bundles in the stem do not have cambia and scattered. It's look like this. What about the cross section of monocot stem? This is the epidermis, and vascular bundles are scattered. In peripheral region, they are very small and high in numbers. Okay, very small vascular bundles. If we enlarge one vascular bundle, it's look like this. It's look like this. No, if this is the xylem, if this is the phloem, no cambium. Okay, no cambium between xylem and phloem. Sclerenchyma sheath can be seen around the vascular bundle in monocots. So, peripheral vascular bundles are very small and high in numbers. When they come into the center region, their size is somewhat large and less in numbers. Okay, anyway, this structure we called as scattered. Okay, vascular bundles are scattered. But in here, vascular bundles in the stem have cambia and arranged as a ring. Okay, it's look like this. If this is the epidermis, vascular bundles are arranged in a ring like this. When we enlarge one vascular bundle, it's look like this. There is the cambium. If this is the phloem, if this is the xylem, this is the cambium. This is the cambium. This is the sclerenchyma cap. This is the sclerenchyma cap. Here, sclerenchyma sheath. Okay, here we cannot see a cambium between xylem and phloem. That is why we cannot see the secondary growth in monocot roots as well as monocot stems. Anyway, xylem and phloem, between xylem and phloem, no cambium, but in here, in dicot stems as well as the roots, between the phloem and xylem, we can see a cambium. Okay, here, in both cases, sclerenchyma tissues are involved to uh, make the vascular bundle. Here, this is the sclerenchyma tissue. Here, the sclerenchyma cap. Okay. So those are the 7 differences. First one, here the embryos have only one cotyledon, fibrous root system, parallel veins in leaves. Flower parts are trimerous. Perianth present in flowers. That means no distinct